Hello, 9th standard. Have a good day. Have a nice day. I hope you are doing well. Today we are going to do video number 25 where we are going to learn economic biology part 3. Right? As we have seen so far, part 1, part 2. Now this is part 3 where we are going to learn about mushroom cultivation, aquaponics and hydroponics. We are going to learn 3 side headings: mushroom cultivation, hydroponics and aquaponics. Okay, and in lessons that you have learned so far, mainly we have learned about different types of floriculture, horticulture branches as pomology, olericulture, greenhouse, floriculture and then landscape gardening. Then we learnt about animal manure where it is as farmyard manure and sheep and goat manure that we have learnt in detail. Then finally in previous video number 25 we have learnt biofertilizers. I hope you remember that. But types of biofertilizers we have learned rhizobium, azospirillum, acetobacter, mycorrhizae and azolla. Are you clear? The next one is page number 276. What we are continuing this part 3, video 25 is medicinal plants as Ayush we say, Ayurveda, Yoga, Unani, Siddha and Homeopathy. These use drugs obtained from plants and animals. And these drugs from medicinal plants are called secondary metabolites. Make a note. It's an important topic to understand. Drugs obtained from these Ayush medicinal plants are called secondary metabolites. Plant produce primary metabolites for their own living as carbohydrate, amino acids, and secondary metabolites are for protection, competition and species interaction. Protection we know, competition in a sense, different plants and animals will require space, shelter and food. So that food what is available we have learnt as primary consumer, secondary consumer and tertiary consumer, is it not? So those consumers, if one eats the other, then there is shortage of first one, is it not? If primary consumer is eaten by secondary consumer more, then there will not be survival for primary consumer. Like that, it's a survival for competition and species interaction. Species interaction may be positive and negative. Positive interaction it is mutualism. Symbiosis we have learnt, that is positive interaction. Negative interaction, these parasites, when they harm each other, it is negative interaction. So here this paragraph, though it is small, you should learn thoroughly about primary metabolites, secondary metabolites, and secondary metabolites are for protection, competition and species interaction. Adu unakku therinjirkanam. Seriya? Example, alkaloids, terpenoids, flavonoids, etc. Then phytochemistry is the study of phytochemicals, which are chemical substances derived from various parts of the plant. That various parts of the plant here given in our page 277. Okay. Now I am not going to read it. You can just go through what you have learnt. Economic biology practicals we have learnt. Under that we have learnt about nilavembu and aloe vera. But aloe vera illai anat padichirkom nama. I hope you follow this. So katrare, first one is katrare aloe vera, then tulasi, nannari, nilavembu, vetpalai, cinchona, then sarpagandha, thailamaram, pappali and nithikalanipu. In all that you should learn the botanical name, drugs obtained and then which part of the medicinal which part of the plant is used as medicine and the disease it cures. So this we will go through. I am not reading, so please follow it up. Sure? The next one, up to this is the introduction that we are learning. The next one is mushroom cultivation. Now the mushroom cultivation, as I said, we have learnt Agaricus bisporus for practicals, isn't it? Now this is the figure given in the textbook also. We have given, it's given mushroom. So mushroom is a fungi producing fleshy fruiting body, that fan like umbrella like structure, no, that is fleshing fruiting body, especially one consisting of stalk with the umbrella cap. Now the stalk is called cap-like structure, pileus, attached with the thread-like mycelium. And mycelia absorb nutrient from the soil. It does not require sunlight for their growth. And this Agaricus mushroom, that umbrella-like structure is a pileus. And then can you see the brown structure below the umbrella? They are the gills. And then it has a stalk called stipe, and the brown gills or a white ring thread where if you observe carefully you can see it, that is annulus, from which the gill start rising. This is the structure of Agaricus species. Okay, and actually what you are seeing, Agaricus is a fungi where the plant body is formed of thallus. The thallus is called mycelium. We have learnt in microbes, fungus, just to recollect the point, that mycelium, it is formed of hyphae, and that hyphae, when it is actually for this Agaricus mushroom growing below the soil, but when it matures, the tertiary mycelium is emerging out as the fruiting body, that is umbrella-like structure, that is called pileus. This is for the structure of Agaricus mushroom. Okay, so now this mushroom can be cultivated commercially. So mushroom cultivation is technology of growing mushroom using plant, animal and industrial waste. In short, it's wealth out of waste technology. This mushroom cultivation is called wealth out of waste technology. It has gained importance worldwide because of its dietary fiber and protein value. Then it comes under the group Basidiomycetes. The gills, brown structure what you are seeing here, that will have the structure called basidium, which in turn producing basidiospores. Okay, so it comes under the class Basidiomycetes, and it is rich in protein, fibers, vitamins and minerals. Okay, there are more than 3000 types of mushrooms. Especially common one here are button mushroom, that is Agaricus bisporus, oyster mushroom, Pleurotus species, padistra mushroom, Valvoriella valvaceae are cultivated, and it takes cultivation 1 to 3 months. I hope you follow. So this is the structure of the Agaricus bisporus mushroom, which is common one found here. Are you clear? The next one, as I said now, mushroom cultivation. This is what we have learnt now. Mushroom cultivation is a technology of growing mushroom using plant. Okay, and plant, animal and industrial waste. It is wealth out of waste technology. It has gained importance worldwide. Why? Because of its dietary fiber and protein value. And it is also rich in protein, fibers, vitamins and minerals. I hope you are listening. The examples are button mushroom, Agaricus bisporus, oyster mushroom, Pleurotus species, padistra, Valvoriella valvaceae. Valvoriella valvaceae, right? And the cultivation, it takes 1 to 3 months. I hope you are listening. Now the stages of mushroom cultivation are composting, spawning, casing, pinning, harvesting and preservation. See the textbook figure here, it is mushroom. For the white one, they just plenty of mushrooms are there. It's, first one, the red color is a single mushroom. The red color is a umbrella shaped pileus. Okay. So what are the process for mushroom cultivation? Composting, spawning, casing, pinning, harvesting and preservation. I hope you understand this. The next one, mushroom cultivation. The steps are, first you have to prepare the place for cultivating the mushroom, and then you have to soak straw in water for 1 or 2 days. Straw na the vacuole சொல்லுவோம்ல, இல்லைன்னா உமி எடுத்துக்கணும். Okay, so soak straw in water for 1 to 2 days. Then mix straw mushroom seedling with supplementary food. Mushroom seedling available, so mix this straw with the mushroom seedling with supplementary food. For the growth of the mushroom, it needs under proper conditions as proper feed and nutrients should be there, ventilation should be there and climatic condition. Then place the mold on the ground. Mold here referring to fungus, right? Then place soaked straw in mold 10 centimeter height and place step 3 along straw edge. Again you have to mix straw mushroom seedling with the supplementary food. Add another layer of soaked sadamari. The vacuole oru layer mela 10 centimeter high la vechinde varanam. Okay, so continue the process to fill the mold and top with the straw. I hope you are following. So நம்ம ஒரு பெரிய ட்ரேல plate மாதிரில soil எடுத்துட்டு first you will keep the straw with the feed, right? Over that you just apply mushroom seedling. Then again you keep this straw like that ஒரு 10 centimeter height க்கு one above the other மாத்தி எதை mushroom seedling இந்த vacuole லேயரையும் மாத்தி வச்சிடுவா. Okay, that is the step for cultivation. Now what happens? First again it is a step explained here. Raw material, paddy, straw. Then chopping na இப்படி கட் பண்ணி வைப்போம் இல்லையா அது. Then fill up the gunny bag. இதில் வந்து gunny bag na இந்த சாக்குப்பை மாதிரி எடுத்துட்டு இருக்கா, right? And that is soaked in water. அப்போ அந்த இதில் ரொம்ப ஊறின்றதுன்னா அந்த நிறைய தண்ணி இருக்கும் இல்லையா, that excess water to be drained. I hope you follow. And then hot water treatment பண்ணலாம், that is for 30 to 60 minutes. Again you will dry the excess water, then you have to dry it up. ரொம்ப ட்ரை பண்ணிடக்கூடாது, கொஞ்சம் வெட்டாகவும் இருக்கணும், சரியா? So this is a prepared food bed, அதுதான் பெட்னு சொல்லுவா. And then pinning the bed, spawn, running. In the steps you have to follow. I hope you follow. Understand this. So this is a step for mushroom cultivation. Okay. So now so far நம்ம textbookல என்ன சொல்லியிருக்குன்னா, we'll come to this next. Just listen. Composting, spawning, casing, pinning and harvesting. Listen carefully. Composting, spawning, casing, pinning and harvesting. Next method வந்து preservation. I hope you are listening. So composting is prepared by mixing paddy straw with a number of organic materials like cow dung and inorganic fertilizers. It is kept at about 50 degrees centigrade for 1 week. இது ரொம்ப சிம்பிளா இருக்கு composting பாரு. By mixing paddy straw, அதான் vacuole, சரியா, with a number of organic materials as fertilizers and manure like cow dung and inorganic fertilizers. Okay, it is kept at about 50 degrees centigrade for 1 week. சரியா? The next step is spawning. Spawn is the mushroom seed. It is prepared by growing fungal mycelium in grains under sterile condition, and spawn is sown on compost. Okay, then compost is covered with a thin layer of soil again, which gives support to the growing mushroom. It also provides humidity and helps to regulate the temperature. This step is very important. I hope you follow. Then next step is pinning. Mycelium starts to form a little bud. The second diagram is white color. Little bud which will develop into mushroom, and those little white buds are called pins. The young plantlet வளர்றது சொல்லுவோம் பார்த்தியா, அது மாதிரி இது வந்து pinning. Okay, then mushroom grow better in 15 to 23 degrees centigrade, and they grow 3 centimeter in a week, which is the normal size for harvesting. அப்பதான் நம்ம எடுக்கிறோம். Then in third week the first fresh mushroom should be harvested. So the 3 centimeter height வந்த உடனே you can do the harvesting, and after removing it has to be preserved. Preservation is also a important step. So discoloration, weight and flavor loss are the main problem during harvesting. And this mushroom is very soft and tender, totally உடஞ்சிரும். So அது இல்லாமல் you should be very careful. And ரொம்ப over dosage இருந்தாலும் there will be discoloration. சரியா? So the following methods are used to increase their life. எப்படி? Preservation can be done as freezing, dyeing, canning, vacuum cooling, gamma radiation, and storing at 15 degree centigrade. I hope you follow. நம்ம இந்த freezing, drying, canningல நம்ம food preservation methodல we have studied. Recollect that point. Nutrition and hygiene lessonல படிச்சிருக்கோம்ல, the same method. Freezing, drying, canning, vacuum cooling, and gamma radiation, and storing at 15 degree C. I hope you understand. The next what you are learning is hydroponics. Hydroponics na actually it is a soilless culture, culturing the plant without the soil. So hydroponics is a method of growing plants using mineral nutrient solutions instead of soil. அதனால தான் இதை soilless culture சொல்லுவா. சரியா? So hydroponics are method of growing plants without soil using mineral nutrient solutions in water. I hope you are listening. The containers are that we will show the diagram, the figure one by one, all these structure. Okay? So now it is meant for advantages. Faster growth rate, less bugs and diseases, better taste, less space, better nutrition, and no weeds. Weed na undesirable plant growing in it. Unwanted plants that grows around a plant are the weed. Okay, so advantages are faster growth rate, less bugs and diseases. Bugs on the insecticides and pesticides சொல்லுவோம்ல, அது insects. And better taste, less space, better nutrition, and no weeds. Now hydroponics, if you ever placed a plant clipping into a glass of water with the hope that it will develop roots, yes, that is a method of hydroponics. So that shows you have practiced in a form of hydroponics. Okay, so hydroponics is a branch of agriculture where plants are grown without the use of soil. The nutrients that the plants normally derive from the soil are simply dissolved in water that is provided. I hope you understand this line. Depending on the type of hydroponic system used, the plant roots are suspended in, flooded with or mixed with the nutrient solution so that plant can derive these essential elements it needs for its growth. Okay, so see the 3 figure, it grows in hydroponic method. So bricks in roof would like proudly walk to future form where we research, develop well-equipped and proven hydroponic system in India. I hope you understand. Then why do we use this hydroponic? Because hydroponic garden produce healthiest crops with a high yield and are consistently reliable. And gardening is clean, extremely easy, requiring very little effort. Either the soil, we have to loosen the soil, plow the soil, and then we have to supply manure and fertilizer, is it not? So it is easy to follow it up. Due to nutrients fed directly to the roots, plants grow faster, and with small roots, plants may be grown closer. And it offers the benefits of shorter harvest time. Easily we can take, right? In general, hydroponically grown garden needs only 1/5 of overall space when compared with the soil garden. And the space occupation is very important. In general, it needs very less space, right? This picture presentation, this particular presentation is not given in our textbook, so just pause the video and make a note, right? Hydroponically grown plants do not come in contact with soil-borne pests and diseases, thus save cost of soil preparation, insecticides and fungicide. Some of the pests the பூச்சி இதெல்லாம், that is soil-borne pest, and it will not affect the hydroponic plants. Okay, so it is said hydroponically grown plants do not come in contact with soil-borne pests and diseases. Thus, it saves cost of soil preparation, insecticides and fungicides. Are you clear? Since the amount of nutrient solution is fed directly to the plant roots, there is no wastage of water due to runoff or evaporation. And hydroponic science allows the people to enjoy locally grown produce and to expand their food production. It also allows to enjoy year-round local produce without the impact of weather, large maintenance expense or delay of supply. I hope you understand. So this particular description, please make a note in your classwork. All the 9th standard, please make a note in your classwork. Only then it is easy for learning. The next one, see the textbook diagram. Hydroponics, its importance, conservation of water and nutrients, controlled plant growth. In deserts and Arctic region, hydroponics can be an effective alternative method. Are you clear? In the previous order, please make a note in your classwork. Then importance, it's given in a textbook, you can just understand, right? So here, this is a explanation for hydroponics. See, it's a soil circular. First வந்து ஒரு drip line இருக்கு பாரு, அதில் plants are grown. I hope you understand. And then the glass container வந்து the nutrient medium இருக்கு பற்றியா, water with the nutrients. இந்த மெத்தடில் you have to follow it up. I hope you follow. Then what is hydroponic system? அதே தான். For example, we know the equation for photosynthesis. What is it? Carbon dioxide plus water in presence of sunlight and chlorophyll, it gives glucose and oxygen. In the equation 6CO2 plus 6H2O, in presence of light and chlorophyll, அது இல்லை இல்லை தெரிஞ்சுக்கோ, C6H12O6 plus CO2. Yeah? And there is no mention of soil anywhere. So that's all the proof you need that plants can grow without it. இது நான் சொல்ல மாட்டேன், it's not correct. From water is come from the soil தானே, அது வந்து this statement is not 100% true to be agreed. சரியா? but still understand soil, without soil plant can also be grown. அந்த மாதிரி புரிஞ்சுக்கும். I hope you understand. So this is for another hydroponics. Importance I already said, I am not going to read it out. Okay, then next one is how does hydroponics work? Other than a 2-3 explanation when I find it is easy for you to understand and learn, I have given. Sorry, actually it's a textbook diagram though, right? First one the nutrient plant, growing plants, and then one vessel இது வந்து first nutrient host சொல்லுவோம்ல, அது மூல அனுப்புகிறோம், then it returns. Then we have nutrient pump and air pump. I hope you understand. Then stand your plants in plastic trough and let a nutrient solution trickle past their roots with the help of gravity and a pump. Are you clear? Then 1 minute, go through this. 1 minute. 12 of 18. Sorry? So this has to be followed. The next one is deep water culture, that is aquaculture, right? So here it is fish tank, then again growing tray, filter, pump. What do you understand? Deep water culture, nothing but aquaculture. Yeah, otherwise aquaponics சொல்லுவோம்ல, that is aeroponics and aquaponics, right? Now aquaponics is a system of combination of conventional aquaculture with hydroponics in symbiotic environment. I repeat, aquaponics is a system of combination of conventional, just understand, the aquaponics is a system of combination of conventional aquaculture with hydroponics in a, I repeat, aquaponics is a system of combination of conventional aquaculture with hydroponics in a symbiotic environment, in which plants are fed with aquatic animals excreta or waste. These waste are broken down by nitrifying bacteria initially into nitrates, later into nitrates that are utilized by plants as their nutrients. இதனை எதை ஞாபகம் வச்சுக்கணும், நெக்ஸ்ட் லெசனில் படிப்போம், நைட்ரஜன் சைக்கிள். சரி, ஸோ thus the waste are utilized and water is recirculated back to the aquaculture system. Now understand, this is again aquaponics, right? Then this is the first one, figure 23.14 is a textbook diagram. See here, aquaculture, hydroponic, both are together forming aquaponics. Are you clear? Aquaponics, fast growing and more sustainable method of food production, and it is a system that combines hydroponics with intensive aquaculture. It is a bio-integrated food production system. Okay, so aquaculture hydroponics சேர்ந்து வர்றது aquaponics. I hope you follow. Then we come across one more as aeroponics in our textbook. It's a system of high-tech type of hydroponic gardening. The growth medium in this type is primarily air. Another aeroponic. Okay, the growth medium is primarily air. The roots hang in the air and are mixed with the nutrient solution. Right? This is done for every few minutes, as roots will dry up rapidly if the misting cycle are interrupted. இதுக்கு aeroponic. அதனால் அதை கொஞ்சம் கேர்ஃபுல்லாக செய்யணும். A timer control the nutrient pump, much like other types of hydroponic system. except that aeroponic system needs short cycle timer that runs pump for a few seconds and every couple of minutes. இது கொஞ்சம் நீ பொறுமையாக படிக்கணும், சிம்பிள் இதுதான் எனக்கு. Okay, the next one is aquaponics. Again, see the diagram. A combination of fish and plant production using aquaculture. A combination of fish and plant production using aquaculture and hydroponics. Aquaponics is moving from the realm of experimental to commercial. So we will learn more about this production system and if it might be right for your backyard gardening or farming operation. I hope you understand something in that. First one வந்து soilless. Okay. Then advantages என்னன்னு பாரு. See the figure here. All of them, they are soilless plant. Reduce labor. Aquaponics uses 90% water than traditional farming and it doesn't require farmland and soil. Fish feed is the only nutrient inputs and it is the aquacultural waste products are used as manure. Okay, and aquaponic, it protects our rivers and lakes and its health and nutritive value is very high and it has a high production rate and it provides income from two separate products. So a lot of current research and improvements are going on for this aquaculture. Okay, so now it is a difference between advantage and disadvantage. What do you know? For advantages, plants are fertilized naturally with the fish byproduct. Growth of plant is significantly faster and easier setup for year around and reduced damage from pest and disease and no weeding or bending down. எதுக்கு advantages of aquaponics. Whereas disadvantage, ஒரு greenhouse we need and it requires technical knowledge of aquaponic system. ஏன்னா இந்த டைமர் பார்த்துக்கணும். Then high startup investment cost because it's for the technology and equipment. Energy usage is rather high with the system and complicated for the amount of fish. I hope you follow this. இது கொஞ்சம் புரிஞ்சுண்டு படிக்கணும் இது, சரியா? நம்ம டெக்ஸ்ட் புக் டயக்ராம் வச்சு கொஞ்சம் புரிஞ்சுண்டு படி. Okay, so now here in this topic what we have done, that is our video number 25 தான் ஞாபகம் இருக்கு. Video number 25 we are learning chapter unit 23 economic biology, where we are learning 3 topics as first one medicinal plants and then mushroom cultivation, hydroponics, aeroponics and aquaponics. These are the topic or the points that we have learned. Okay, so please go through the lesson and then again as I said initially you should know about the another ஆயுஷ்ல plant drugs are extracted. அதில் வந்து you should know about secondary metabolites, அதோட purpose, why is that secondary metabolites. I hope you understand. And then this page 277, drugs derived from medicinal plants. I am not reading, but you should know to understand the botanical name, drug, parts used and disease cured. Okay, just for a few seconds listen pannu. Drugs derived from medicinal plants. 1 minute. Drugs derived from medicinal plants given in our page 277, where first name Tamil name வந்து கத்தாழை, botanical name aloe vera and drug is anthraquinone. The name மட்டும் படிக்கிறேன் so that you will know to pronounce. then disease cured, heal wound, skin disease, cancer, psoriasis. Then tulasi, osmium sanctum, nannari, hemidesmus indicus, nilavembu, andrographis paniculata, vetpalai, wrightia tinctoria, cinchona is Cinchona officinalis, then Sarpagandha, Rauwolfia serpentina, then Tylamaram is Eucalyptus globus, then Papil, Papali, it is Carica papaya, Nityakalani plant, it is Catharanthus roseus. I repeat, aloe vera, osmium sanctum, hemidesmus indicus, andrographis paniculata, wrightia tinctoria, Cinchona officinalis, Rauwolfia serpentina, Eucalyptus globus, Carica papaya, Catharanthus roseus. Okay, so these are the medicinal plants that you need to learn. I hope you understand. So this is our video number 25, chapter unit 23, economic biology. Okay, I hope you are listening properly. So I have given 2 assignments here. You have to write the assignment. I repeat, whatever the assignment given to you for this lockdown, please copy. You have to submit when we meet. Okay, so we'll continue in the next video session. Have a good day. Have a nice day. Thank you.